Hello, everyone. So we've just started the session, and I'll be talking about what you can learn about robustness of studies in large-scale replications compared to meta-analyses, with a focus on developmental science. But I'm sure that even if you're not a developmentalist, you can take some lessons home for you and your own work. Usually I like to do this in an interactive way, but to make life easier for the organizers, let's keep questions until the end. If you have clarification questions, I'll try to monitor the chat, so just put them in the chat when something's unclear during my talk. But otherwise, let's have a very, very lively, I hope, discussion at the end. And then we can also jump back through the slides, of course. So let me start with how I think about replication, because we're all about replications this week, and hopefully more than just this week. There is an original study, and any pair of studies compared to this original study, any second study lives along a continuum that's either close or far away from this original study. So for example, if you run an exact replication, right, there's sampling error. But if you alter things, you're very quickly into broad, varied replication, conceptual replication, or even generalization territory. The more you alter, the further away you go. So this is how I think about this, and this helps me thinking about meta-analyses and large-scale replications. So you don't need to adopt this framework, but this is what I usually use to talk about replications in general. So let's quickly change pace and talk about infant studies. Infant studies are always great because you get to work with these very cute human beings, but they are very noisy. So as soon as you go into experimental work with infants, you have many sources of noise because infants are very difficult to recruit. You can't just ask them like when they're in the cafeteria, as you would do with maybe adult studies, where they would just quickly want to pop over for a short eye tracking study. No, you have to, of course, also invite their parents. You have to inform the parents that this is okay. Having an infant is a very exhausting thing. This alone is a hurdle that infant researchers have to contend with. Infants are also really difficult to test. You can't just instruct them to do what you'd like them to do. You can't just ask them, okay, click on the picture you recognize, or tell me whether this sounds familiar or whether you had that before. So we only have to rely on implicit tests, and we have to hope that they test what we are testing, or what we are looking at. And then they're also very variable. So no two infants are alike. Anyone who's had to hang out with more than one infant probably knows that. Even before birth, we can see differences between whether babies are very active or very concentrated, easy to amuse. So they differ on many dimensions just between any two babies. But there's also the age factor. So a young infant, of course, is very different being from an older infant. Maybe they can already sit. Maybe they can already say their first words. So that's already a huge difference between infants. And of course, there are cultural differences and probably many more things that I haven't even thought of yet. So it's, all these sources of noise coming into infant studies. So we are contending with a small sample, with indirect methods and a very invariable population. So one solution is, of course, to team up and to run large-scale replications. And for that, there is many babies. I just assume that you haven't heard of many babies yet. If you have, just quickly browse the website, or I'll still quickly introduce. So the common theme is early development. And it's a network of developmental scientists that runs large-scale collaborations. I see something in the chat. Yes. Thank you, Lucas, for adding the website. Yes, perfect. There's a lot of information about many babies also on the website. So I'm just giving you the highlights. It's a quick summary. So the goal is to promote open sciences and best practices. And what we do is we run consensus-based replications. So that's maybe something to really emphasize in this context, that replications aren't just, we take one very influential study and then we try to as closely replicate it with a larger sample as possible. But we take a question, and I'll give you an example in a minute, that we then discuss with experts who've been working on these questions, maybe 20, 40 years. So really people who've dedicated their life to these topics. And we also junior researchers. So I was a very junior researcher when I started with many babies. I just had finished my PhD. So both these experts and junior researchers with maybe new ideas come together to come to an agreement how to test a specific question. That can lead to very heated discussions, that can lead to new designs, that can lead to insights just based on these dialogues, which is very, very interesting. But it's different from other replication projects that you might be more familiar with, where one team of researchers completely designs a study and then invites people for data collection. So that's something I really want to hammer home. And what we're addressing as both theoretical and methodological open questions. Because methodological questions are relevant for our theories, right? Because we need to know what our methods actually do, how they tap into infants' cognition. So I hope that's clear so far. So many babies lives on this continuum, more in the broad replication, maybe even conceptual replication land compared to an original study that we might, of course, refer to. So how does one specific project get designed? There's still a small group of leads. There needs to be a group of people that is responsible. And they propose a question to the governing board. And they benefit from, all that we've learned in the past 10 years almost. So Many Babies is almost 10 years old now. The first, so we've started with like an official structure in 2017. That's also when the first paper came out. So we've been at it for 10 years. We've learned a lot. And we have a code of conduct, just like Ford. And we have agreements on data use. And that's, again, this crucial difference from some other replication projects, that really the openness starts at the design stage. So we've just started 2 new Many Babies projects, and they had open calls when it was like, okay, this is the broad question we want to test. We've convinced the governing board that these are important questions we want to test. Now give us your input. And that's also, of course, very good for buy-in from the community. So this is how Many Babies is structured. A project usually it has a main project, and then it has spin-off projects. They might collect parallel data, and then there are secondary analyses. And what I'll be presenting here is a secondary analysis of collected data from the main project. So this is still part of Many Babies. We still live in the Many Babies universe here. And this is Many Babies 1. It's complete. This is just a screenshot from the website. But you see there's this one numbered project, Many Babies 1. And then there are all these spin-off projects where we've realized that we didn't have any infants from the whole continent of Africa, for example, tested, which is ridiculous given how huge it is. But of course, we know how biased our data collection is, how money plays a huge role in buy-in to replication efforts. So there's a specific project just to test African infants, to see how we can support researchers who might not have the resources, who might not even be in the same research traditions as we are. So I think that's just one example of a spin-off project. And further down, the second to last is the comparison to meta-analytic data, which is a secondary analysis. So you see there are a lot of things going on. Oops. That, of course, didn't work. So the research question is, do babies prefer infant-directed speech? My picture's broke here, I'm sorry. But what it's supposed to show is a father speaking to his child. And when we speak to our children, we use a different kind of pitch. So American English is very famous for doing it in a very extreme way. When they say, look at this baby. I'm German. Apparently, I do it less when I talk to my child. So I would more say like, "Oh, schau mal." But I would still use a different kind of register, different words. So I speak in a different way to a baby than I speak to an adult. For those of you who aren't interacting with babies, think about how you talk to a small animal, like a puppy or kitten. That's actually related. So we change how we talk based on who we're addressing. And what people have, yeah, then realized is that babies really like this way of speaking. We don't really know why and what the purpose is, but babies like it. That's at least a proposal. But yeah, it's still an open question. Do babies, is this something that generalizes? Because it's such a foundational assumption in developmental science. We design our studies that way. We try to address babies that way to make them cooperate with us. But if they don't like this way of talking, then we shouldn't do that. So it's really foundational. So what did we do? We launched a call, we designed a project, and had very, very heated discussions about how to test for this preference. What we ended up with were 69 participating labs in this main project. So that doesn't contain, for example, the African data. We collected data in 16 countries, including Japan, Australia, South Korea. So it is a global effort. And we tested over 2,000 babies, which for an infant study is huge. I think that's the biggest experimental infant study I'm aware of, at least. So there are these cohort studies, but this is like on a single question. I don't think we have that many babies. And we have 149 authors. We published in AMS, which you might be familiar with, which was yeah, one of in its early stages when we published and was really, really supportive in getting this paper out. And yeah, we did the whole registered report thing, of course. So what did we find? There is a main effect, and it's there. It's positive. So positive means babies do prefer infant-directed speech. Negative would mean babies prefer adult-directed speech. So yes, there is an effect. This is effect size in terms of Cohen's d, standardized mean difference. For those not familiar, we can have a little detour maybe in the question session. I'll just presume that you're sort of familiar with effect sizes because we also need it for calculating power. So yeah, we did find a main effect. It's there, it's real. That was a huge relief. That's not a huge effect, but it's good for infant studies. It's a substantial effect. You don't need hundreds of babies to find this effect reliably. But there is this interesting phenomenon in developmental science that we constantly change our methods. And one of the secondary questions of many babies one was, does that matter? And we did find a clear method effect. So this figure is super overwhelming, I know. What you should focus on is that there are 3 different colors, which are self-assigned methods. So labs could choose the method. And all of them have a different effect size. So this little star in the bottom, I don't know whether you can see my mouse, but I'll just try to, yeah. I'll use a, but yeah, there's a little meta-analytic estimate. And this little diamond is different across the 3 methods that were used. And these are really methods that are super common in infant research. They are thought to be interchangeable, but we find that they matter. And that's really, I think, a very, very important finding because we assume the stimuli were the same. We assume the underlying preference, the strength of the preference doesn't matter. So there needs to be something happening between baby saying, oh, I like this. We're not saying, of course, and our ability to measure the strength of that response. And of course, you see there's huge variability. The size of the dot reflects the number of tested infants. So how sure we are, the bigger, the better. I don't want to judge, but of course, effects are more reliable, more robust when they have more infants. You can estimate the effect better. But you see, even yeah, lots of infant tested might give the opposite effect. So we see lots of variation in single studies, but a robust meta-analytic estimate. So this is also, yeah, basically a linear model that gave us these effect sizes. Something else that we find is an age effect. Pew, that's also really good because babies change as they age. We use the same stimuli across all ages. Usually infant researchers would adapt their stimuli to the different age groups they are testing. We have these 4 age brackets. I won't go into details, but they are all up to 14 months. They're really small, right? So here they can't even sit in the younger age bracket. At 6 months, they usually start sitting, and these oldest kids, they can actually walk and run away, which is really, really hard for infant research too. But what we find, yes, there is an age effect. What we also find is that we find language effects because our stimuli were in North American English, because that had been attested to elicit strong effects even in babies who do not grow up exposed to North American English. For example, babies growing up learning Cantonese. So that's why we ended up settling on these stimuli. And there is a difference in how much babies react based on their own native language. That's really, really cool, I think. So we did learn a lot. We have a main effect. We have some secondary insights. And again, I have some image issues with my Google Slides, sorry. And let's compare that to a meta-analysis. So there is a published meta-analysis out there that's gray literature, so-called gray literature. So it's not gone through a proper peer review. But it's a meta-analysis that we could use and that we built, also actually estimated our power on when we planned many babies, and they reported an aggregated effect size of 0.67, which is a bit higher than 0.35, right? We're not even in the confidence interval, but they have way less babies included. And what is interesting, what you can't see right now, is that they actually have separated their meta-analysis into which kind of stimuli you use, because people across studies use different kinds of stimuli. Some people use artificial speech. That was way before we had AI, right? We're talking about the 80s here. So they had these speech generators that are very mechanical, and they sound like you're talking into a box, into a tin can. Of course, that's to remove any personal characteristics of the voice. Babies like this least, and then their naturalistic stimuli, and in this meta-analysis, baby like that most. And that's where many babies also lies. So if we were to subset the meta-analysis to the part that's closer to many babies, we would even expect a higher effect of around 0.8. So the difference is even bigger when you try to match the meta-analysis to the replication. There's also an updated meta-analysis, and that lives on MetaLab. So I have to now tell you about MetaLab, which is my pleasure. MetaLab is another fun project that addresses some problems with meta-analyses, which are, of course, collections of replications. So they should be interesting to all of you. Usually when we do a meta-analysis, and I would actually like to know whether any one of you has done a meta-analysis, whether you've gone through the very painful process, because it's a literature review plus, to say it quickly. So it's a high workload. Once you're done, the data are closed and static usually. So what do you do when there's a new study on the topic coming out? Do you run a whole new meta-analysis? Do you have to do the whole work again? Especially if you don't have access to the closed data. If you're interested in a different variable that the original meta-analysts haven't coded for, do you have to run a new meta-analysis? It's usually intransparent what happened, which decisions were taken, how you select studies for inclusion, exclusion, so how you actually arrived at the effect sizes, which regression model you used. All the things that are basically wrong with experimental literature can be found in meta-analysis as well. Analysis, decision, exclusions, random effects. The format is idiosyncratic. And of course, there are things like publication bias and other biases in inclusion and exclusion. So all the things that we're struggling with can be found again. So what have we come up with to address these issues? It's so-called community-augmented meta-analyses. Usually call them karmas. So try to remind yourself that this is what karma stands for when it comes back later. So it's shared, and you share the work and the resource. It's open and dynamic. So if there's a new study, you add it. if you have a new variable, you code for it. It's as transparent as possible. So MetaLab completely lives on GitHub. You can clone it. You can completely look at all the different decisions we've made. We've also tried to document our code and our process. Basically, yeah, we're trying to address all of these issues. And again, it's broken. So let's skip that. So on MetaLab, we have 117 conditions, and we have an overall effect size that's actually more similar to the one on that we found in many babies. So why is there this updated meta-analysis? Of course, we've added new data that has been published since 2012. And we actually discovered, while we were working on this project to compare both the large-scale replication and the meta-analysis, that the original meta-analysis had some issues. As I promised, we have the same issues in experimental literature as in meta-analytic literature. There were effect sizes that were just not correct. So it's easy, of course, it's human error, right? You try to extract from a paper from 1983 mean and a standard error, and then you mix it up and you put it in the field for standard deviation, and the whole formula changes and your calculation is wrong. That can happen, right? We also disagreed with some decisions they made. That's all very detailed and documented in high detail, actually. There's a long list there's a whole spreadsheet of where we disagreed and why we changed things. And they were also not as consistent as we'd liked in coding so-called moderator variables, so things that describe how the study was run. So for example, about the methods used or the stimuli used, right? We want to have a consistent way of coding these things. But yeah, so things that live in MetaLab that are, on this replication space, compared to one original study that started the whole thing, that's usually the oldest study, a meta-analysis lives on this much broader spectrum. So there are some very close replications. In infant research, I would say they are very rarely exact replications, because to make it publishable, you always have to add a twist. But it goes really broadly to what I would almost call a generalization study. And that's just to give you an idea of how much these studies differ. So what we need to do now, if we want to compare these two, we need to find a way to contend with these differences, to weigh these differences, or to find different sources of variation. So what we're doing is, as I mentioned, we code everything that we think is theoretically relevant in both sources of data. And for that, you need knowledge on the domain that you're working in. So you need really experts from the specific research question. And then you, of course, have to check, do I have the data to actually analyze this? Is there something that's present in both data sets? Is the distribution somewhat that I can analyze it? Does the model converge, right? The classical issues. So what we have here are the candidate moderators, among which are, of course, method and whether the language was native or non-native. So very similar to what I presented earlier for many babies. The other questions, and so in red is always many babies, and you see that for basically apart from the top level, the top two categorical moderators, many babies is just on one dimension there. So many babies has always made a decision that we use naturalistic stimuli. We don't use filtered speech. We don't use simulated speech. We have never used the mother. There's just nothing happening there. We didn't go and record always mom's speech to present that to baby. We have no audio, no video recording. We have never checked whether babies smile more when they hear infant-directed speech. That's affect, so what's coded as dependent variable. We only looked at whether they prefer it. And our main question was, of course, is there an infant-directed speech preference? So we can't say, okay, this is relevant data, but the main research question was a different one. So right, but they're all theoretically relevant. So what we did is start with that set, and then we ran our meta-analysis. And this is really this naive meta-analysis that takes all the effect sizes and just throws it into a model. And this is, just to be clear, it's always using the new data set, because the other one was just, yeah, in our opinion, not of high enough quality. This figure can be found in the supplementary materials of the publication that I've here linked here. And I'll just put the DOI, well, I can't, because I can't copy from the slides. I'll put it in the chat in a minute. But yeah, so this is what it looks like. And what you can see, of course, is that the variance is much higher for the meta-analysis compared to the replication study. And this is an ugly result table. What I want to show you, though, is that we have one meta-analysis that was moderators plus all the effect sizes, and then we have allowed for an interaction of the moderator with the data source, because the first model wasn't giving us any significant moderators. We couldn't explain any additional variance with the simple moderators. What happened then was, apart from age, of course, so there's an age effect. What happened is that there is an interaction between the method used, which we saw has a strong effect in many babies. So that's yeah, that changes things here. It's the bottom line, and we find an effect of source by age. So an age effect differs across the meta-analysis versus many babies, which I think is very interesting. And let's just visualize that. I think that's much more useful. So when you look at age, this is the top line. It's flat in the meta-analysis. Wow, right? In many babies, yeah, we did find this age effect. It's basically what I presented to you earlier. But in the meta-analysis, it's flat. Why might that be? One theory, to just skip ahead, is that we find that studies, as I'm kind of previewed earlier, adjust their stimuli to the age of the population, because the way we speak to a 3-month-old is different than the way we speak to a 2-year-old. Because a 2-year-old can actually talk back. So that might be part of the issue here, that people adjusted to the tested age. And so the preference, while still present, changed over time, or didn't change in its size. And the other thing is that method basically flipped around in the meta-analysis versus the replication. So this method, that's this very cryptic HPP. For infant researchers, they'll know, and if you don't know, you don't have to, because I think it would take 15 minutes to explain it. This HPP here, it has a higher effect size in the replications and a lower effect size that actually includes 0 in the meta-analysis. That's the bottom left, by the way, that's B. So that's surprising. That's why we find an interaction there. And then we have test language, which I think didn't get significant, but it's there for completion, because that was one of the main moderators. But I think it's interesting that one effect is basically flat versus present, and another effect is even flipped around in both data sources. So that requires more reasoning about how these data were collected, what happened, how method was assigned, how people choose method in infant testing, versus how methods tap into our into babies' cognition. So what I learned from this, we're nearing the end of my part, because I actually want to know what you think and what you learned, is, in this case, wow, we're good. There's evidence for both, in both sources of data, in the meta-analysis and in the large-scale replication. But still, individual published studies, also individual studies in the large-scale replication, show the opposite effect. So if you run one replication study of a source study, it might still be possible that you switch signs, that you show the opposite effect. That's maybe one learning that's super general. I mean, as statistically we know this, but it's nice to see, and I think it's very reassuring that and also gives you some perspective maybe on your own replication results. And it's always more complicated than you think to pre-register. That's I think a general truth, at least for infant studies. The last most informative analysis where we allowed the interaction with source actually was an exploratory analysis. I should have told you, I know, but I thought this is a nice reveal here. So that's the one that told us most. We didn't expect that. We didn't expect that things would go different directions across data sources. We couldn't foresee that. And I think if I were to run it again, I would always include this analysis. It's obvious in hindsight, right? And it's very important to consider how large-scale replications and meta-analysis sample from the space of possible studies. So yeah, that what it allows you to look at, what it allows you to, or what it doesn't allow you to look at, but also what might be sources of unexplained variance. And actually, yeah, usually neither are completely random. Of course, there's always sampling error, but everything else, like the choice of method, the choice of stimulus, is not a random assignment, so you can't treat it as random factor. Yeah, we still need a better understanding of the task, but there's a Many Babies project for it, and they just started data collection, so I'm really, really happy. And of course, we need to document much more, because a lot of things that I was interested in, we couldn't code for. I was very curious, for example, whether things were coded on the fly or whether they were coded by looking at videos. And for everyone who transcribes, for example, interviews, there's a huge difference between the two, right? And much more room for error when doing things on the fly versus offline with all the time in the world, with coming back to it. So that's usually not documented about infant studies. And ideally, we also add everything to community-augmented meta-analysis. But that's, yeah, me as someone who is part of the Meta Lab team, I of course have to say that. But there's also an additional take-home for you, apart from these insights about meta-analyses versus large-scale replications. There is also a community-augmented meta-analysis platform. If you don't do developmental science, and I just want to advertise you for PsychOpen Karma, which is part of the Leibniz Center. So they're part of PsychOpen. You might have heard of them. They're really cool. They give you a lot of resources, and they are really doing not just the kind of meta-analysis I showed you. They also do network meta-analysis that are a bit different. So they are even broader in their domain. And there's also an educational one, because I did hear that the next talk is actually about educational psychology, right? And so I'll add that later to the slides before I upload those to Zenodo, and I'll fix the pictures, of course. But that's actually all I had today. And I'll just, I'll stop sharing my screen so we can actually have a nice chat now. And I'm happy if you, ask your slides, and then if we need to jump back into the presentation, let me know, then we can talk about specific things that you have, that you have questions about.